Well, let's start. Good afternoon and welcome HD community. Thanks for joining us. I'm Professor Luciana Munhoz, a researcher from São Paulo University, the coordinator of the Scientific Committee of Brazilian Huntington Disease Union House, and today I'm representing the Brazilian Huntington Disease Association, or ABH, which is sponsoring this webinar. Thanks, ABH, and a special thanks to Mr. Pedro Henrique Palma from ABH for organizing everything. Thank you, Pedro. This afternoon, we are presenting the research that won a prize at MENA Conference for Rare Diseases, named as Pharma Intelligence and Huntington's Disease, Unveiling New Treatment Horizons, developed by Professor Rodrigo Araldi and Professor João Rafael Dias Pinto. Professor Rodrigo Araldi, who is the speaker, holds a PhD in biotechnology at University of São Paulo, with a postdoctoral training in molecular biology at São Paulo Federal University. He has been investigating neurodegenerative diseases for more than 10 years, and he is the founder and the scientific director of BioDecision Analytics. Professor João Rafael Dias Pinto holds a PhD in sciences from University of São Paulo with postdoctoral training in Pharma data analytics at São Paulo Federal University. He worked at the Department of Earth, Planetary, and Space Sciences at the University of California, United States. And he is the professor in MBA program in Business Analytics and Big Data. He is the co-founder and the Artificial Intelligence Director of BioDecision Analytics. Professor Rodrigo Araldi will initiate the presentation, and you can send your questions in the chat. We will choose some of them to answer in the final 10 minutes of the presentation. Professor Rodrigo Araldi, welcome.
Thank you, Luciana. Thank you for everyone. It's a pleasure to stay here in this webinar. And before I start this presentation, I would like to share here my gratitude for Associação Brasileira de Huntington, or Brazil Huntington Association in English, and particularly Dr. Luciana, who is being coordinator all the discussion with Brazilian teams, international teams, describing our data that is very important for all scientific community and even for our patients, describing new horizons for the treatment of Huntington disease using pharma intelligence. So I'd like to start this presentation not talking about Huntington disease, because as you know, here is a very common issue for everyone that is taking this presentation, but describing a little about the history of the disease that is very related to our study. As you know, the Huntington disease was first reported in 1872 in Long Island, USA. However, the cause of the disease was only discovered 120 years later after this first report. In an international consortium about the disease was discovered the Huntington is caused by one mutation in one specific gene. However, besides this data, today we have more 28,000 publications in literature about different topics involving Huntington disease. And interesting more than this, we have more, or specifically, 256 clinical trials already done or terminated, most of them terminated or have been in complete studies, describing potential studies for therapeutic drugs. In terms, the most part of these clinical trials involved the investigation of drugs, with more than 2,000 subjects enrolled in these studies. And if you consider the high amount of publications and even the high amount of clinical trials already done for Huntington disease, we have one question: why we don't have any specific approved treatment able to postpone the disease, the natural history of the disease, and avoid the neurodegeneration to occur? We have some few drugs that are approved internationally, and the benefits of some of these drugs is very questionable. We have some points to be discussed because you know that these drugs only offer benefits related to some parts of the disease, controlling the chorea, for example, or even promote some points of motor scale function of our patients. So the main question for answering these points is why there is no specific treatment for Huntington disease with proven efficacy. And we can answer this question by two different points. The first of one, the lack of prognostic targets that we call in science biomarkers. Biomarkers are molecules to provide the efficacy evidence of these investigational products or drugs in these clinical trials. And second, due to the lack of therapeutic targets that can be pharmacologically modified. In terms of drug development, the discovery of these therapeutic targets is a very important issue to be resolved. We cannot develop one drug if you don't know what is the target that I need to modify using this specific molecule. But before we start this presentation and show our data, we need to discuss what are these drug targets and biomarkers. As you know, our DNA is responsible to encode thousands of molecules. These first molecules that are codified by our DNA, we call it transcripts. And part of these transcripts will be encoded in proteins. For any type of disease, we have one difference between the level of expression of different types of genes. This occurs even in Huntington disease, for example. So you can compare and analyze these genes in order to identify what is the genes, transcripts, or even proteins that are altered in patients with some specific type of disease. At the moment that we identify these genes that are differentially expressed, we can investigate how many or who are these genes that can serve as potential drug targets or biomarkers. Drug targets are molecules, typical proteins, associated with a specific disease process that can be addressed by a drug to produce a desired therapeutic effect. Is our target from which our drugs can act. Whereas biomarkers are measurable indicators of some biological state or condition, with numerous clinical applications that provide a unique information about individuals' health status. For the drug development, we need all of these things. We need to be a drug target and even a biomarker. Drug target to develop a drug, and a biomarker to monitoring the effect of this potential drug to prove the efficacy of this drug and promote the benefits that we hope that this drug can develop in our patients. And how we can understand a disease at a molecular level. There are many and different types of techniques that we can use for this purpose. But basically, what we've done in science is compare specific tissues of patients that have one specific disease and comparing all these characteristics of these tissues, including the DNA, including proteins, and even the transcripts, with disease-negative individuals that we generally call controls individuals, or only controls. When you compare patients with controls or cases and controls, we have a potentially technique to identify these drug targets and even these biomarkers. When we analyze these results, we can observe different types of results. The first of one, we can observe genes that are equally expressed between patients, for example, with Huntington disease, and our control individuals. These genes that are equally expressed, unfortunately, don't serve for us as a drug target, either as a biomarker. However, we can find genes that are overexpressed, or as you call, upregulated in patients with Huntington, but is less expressed in control individuals. These genes are called differentially expressed and are upregulated. For this reason, these genes can serve for us as a potential drug targets and even biomarkers. By contrast, we can observe an opposite effect. We have genes that in patients, for example, in AD, can be less expressed than in our controls. By being differentially expressed in our target disease, we can explore these genes has the same potential as an upregulated gene, this gene that now is called downregulated can also serve to us as a potential drug target and or biomarker. So, using this approach, what we've done to understand the disease, or Huntington disease? We compared samples from the prefrontal cortex. Prefrontal cortex is a brain area that is very well stated in literature that is affected by Huntington disease. However, unfortunately, we cannot collect brain tissue samples from live individuals. For this reason, the samples can only obtain from post-mortem individuals, making it very difficult to make this type of studies. Because when you're talking about Huntington disease, we are discussing about one rare disease. For this reason, when you need to collect post-mortem sample, we include another difficult to obtain the samples. These samples, once collected, need to be subject to several laboratory steps. One of the most important is to obtain the RNA. RNA is the same that I first called transcript. Is the product that are encoded by our DNA. Is the product of our genes. These RNAs can be subject to RNA sequencing using high-throughput technologies, with a cost about $350 per sample. However, each sample from each individual that we are analyzing generates a big data, or about 16 gigabytes of data that require different types of bioinformatic techniques to be analyzed. This bioinformatic techniques is very laborious and have a cost of about $200 per samples, only considering the costs of analysis, or about $9 per working hour. However, fortunately, we have public repositories such as the Sequence Read Archive, that is an American public repository, in which scientists around the world can deposit your studies and your sequences from different patients. And with us, we can obtain these samples without no cost and don't need to access new samples from post-mortem individuals, making it possible to analyze and reanalyze extensively all these samples in order to obtain specific results using new technologies, such, for example, pharma intelligence that we are discussing in this webinar. Based on this, we collect in this public repository of SRA about 315 samples from our individuals that corresponds to more than 2 terabytes of data of individuals with and without Huntington disease. Considering the costs of RNA sequencing and the analysis, they still would have estimated cost of about $109,000. But since the data are publicly available, we save a lot of money. But with this, collecting all these samples from the SRA, we conducted the largest study of AG already done. In our study, we analyzed 353 samples, from which 146 samples were collected from patients with Huntington disease, making this the largest study ever made. Considering all the literature, many other studies was previously published using the same approach. However, all of these studies was investigating about 15, 7, 20 samples of Huntington patients. Here, we take all the samples together to make one sample enough to obtain satisfactory evidences, identify potential drug targets, and even biomarkers that can help us to develop new treatments. Based on this, we compared our control samples from individuals without Huntington with individuals that carry the AG mutation. However, analyzing our control group, we understand the human population, as even all population in our biology, is very heterogeneous. For this reason, we need to identify what is the correct controls for our study. In previous study, we demonstrated that the age of these patients affect directly our results. If you use control individuals older than patients with Huntington, with this inappropriate usage of these unmatched control individuals can lead to the misidentification of these potential drug targets and biomarkers. For this reason, we used one statistical technique very commonly used for 2 decades that it can exclude samples of unmatched controls, obtain an age-matched control that was compared to our patients with Huntington. If you compare the mean age of our patients with Huntington is statistically similar to the control samples. With this, we exclude patients or individuals, control individuals that are older than our Huntington individuals. Comparing this group of Huntington and controls, here is, only to finish this point, this is our publication demonstrating how the age of controls, if was not properly selected, can affect our results. And investigating all these first published papers that aim to identify these potential drug targets and even biomarkers, we observed many of genes that unfortunately do not serve for us as drug target and even biomarker. And this occurs because the misidentification of these potential targets due to inappropriate use of control samples older than individuals with Huntington. Details of all this study that describe how the age of these controls affect the identification and the accuracy of the RNA-seq analysis can be found in this publication in Frontiers in Genetics that was published about 1 month ago. So we understand that control the aging of our control individuals per se is not enough to lead a correct identification of these drug targets or biomarkers. We need more than this. Because, first of all, we have many different approaches to analyze these samples in terms of bioinformatic techniques. Who is the best? There is no gold standard technique to analyze this bioinformatic process. For this reason, we developed a novel and innovative solution to identify this potential target for all types of disease, including rare disease, such as, example, Huntington. Our model combines different types of technologies and counts on with specific algorithm that is a bioinformatic technique that combines all of these bioinformatic tools available and commonly used by pharmaceutical companies and even in science. And also, we included AI or artificial intelligence algorithms. Artificial intelligence in this context, I am not talking about artificial intelligence that you can find in Amazon or even in Google Assistants. I'm talking about statistical techniques very well consolidated by literature. And with this artificial intelligence that comprise our technique, we can combine genetic data from these patients with clinical pathological features, or I'm here discussing about signs and symptoms of the disease that can be combined with these genetic characteristics to identify these potential drug targets. This is the first time in literature that all of these clinical pathological features were connected with our transcriptomic data or genetic data of our patients. Using this technology, we identified then from the more than 62,000 products that are encoded by the human genome, more than 26,000 are expressed in the prefrontal cortex. However, interestingly, from all of these expressed genes, half of them, or more than 13,000 genes, are differentially expressed in patients with Huntington. So here it's very clear for us that Huntington promotes genetic changes and modify the expression of these genes, and as a consequence of proteins that lead to disease. However, this is well stated for us in literature. We know this and we hope that we expect that this occur. However, when I am demonstrating that more than 3,000 genes are differentially expressed in the brain of patients with Huntington, I have one question. How many of these genes are potential targets to develop one drug or can serve as a biomarker to provide evidence of the benefits promoted by one drug? It's very difficult to identify these specific genes in a high amount of genes that are differentially expressed. Today, in literature and science, what is commonly performed in one case like this? We identify potential target genes using different approaches, but all of them are very subjective. And these approaches, it's only based on characteristics that are imposed by the investigator. Here, use artificial intelligence to identify these potential targets according to signs and symptoms of the Huntington disease. And with this, for the first time, we identified about 400 target genes, specifically 394 target genes. These target genes are very important, and all of them was not previously reported in any other study. But how we can prove that we are correct? Here we are breaking the science. We are demonstrating new horizons. But the main question is how we can prove that these target genes are correctly target genes and not only identified by one artificial intelligence. For this reason, we return to our samples and investigate the expression level of this biomarker in our patients. And interesting, we observed when this is one type of graphic commonly used in for this purpose, in which we can observe here samples from patients with Huntington, and here samples of control individuals. It's very clear to observe by difference of colors between this area in this graphic that the genes that we identify as potential target by being upregulated or increased in expression in Huntington individuals is in fact increased. Here in blue, we demonstrate genes that are overexpressed. And here in red, genes that are less expressed in relation to our patients with Huntington disease. By contrast, we confirmed that those genes that were identified as downregulated is in fact downregulated in comparison to our control individuals. Here we demonstrate that all of the genes identified by the artificial intelligence are accurate. We have specific genes that are differentially expressed between controls and Huntington patients. But only these results is not enough to prove that these genes are good for us as a potential drug targets or even biomarkers. As previously talked in this webinar, I discussed that to be considered a potential drug target or biomarker, it's very important that this gene encode the protein and that this protein needs to be related to our disease. For this reason, we use several bioinformatic tools to understand what is the effect that these genes promote in our patients by being differentially expressed. Interestingly, when we analyze the effect of these genes, we noted that all of them are involved in processes such as neuroinflammation. As you know, the Huntington disease promotes a very well-known state of neuroinflammation. These genes modify the transport across the brain-blood barrier. This is another important issue for the disease. Promote metal-ion response, affect cholesterol metabolism in the brain. leading to axon guidance alteration, resulting in the reduction of the axon, that is the part of the neurons that connect one neuron with other. We have these genes we also observe that are enrolled in neurotransmission alteration, leading to behavior or neuropsychiatric symptoms of the disease. All of these results are in agreement with the pathophysiological mechanism of Huntington disease, providing evidence that reinforcing that the genes that we identify as potential drug targets serve accurately as a drug target for us. Interesting, we also observe that these genes are differentially expressed not only in brain area that we analyzed in this study. Using public database from different types of studies, international studies, we identify that these genes are also differentially expressed in other brain areas that are involved in Huntington disease progression, but it's also related to other neurodegenerative disorders, such, for example, Alzheimer's disease, Parkinson, amyotrophic lateral sclerosis, frontotemporal dementia, and others. This is a very important aspect because if this data is correct, the drug targets that we are describing for Huntington can serve for the treatment of other neurodegenerative disorder, bringing new horizons not only for Huntington patients, but also for patients with Alzheimer, Parkinson, and other diseases. To evaluate this aspect, we refined our results and we investigated how is the level of expression of these genes, these target genes, in other diseases. And you confirm it, as you can observe in this result, in this graphic, we are demonstrating that these same targets that we identified here as differentially expressed in Huntington, it's also differentially expressed in other diseases, such as Alzheimer, Parkinson, Friedreich ataxia, that is another type of neurodegenerative disorder, dementia, sclerosis, autism, bipolar disorder, schizophrenia. These results reinforce our previous data, demonstrate that the target gene is identified by our artificial intelligence is a potential drug target and biomarker, not only for Huntington, but for a set of neurodegenerative disorders. However, the main focus of our study is Huntington disease. And for this, we try to identify if our genes are related to symptoms and signs of the disease, and if the modulation of these genes using drugs, for example, could postpone the progression of Huntington disease. For this reason, we investigated how many of these about 400 target genes can be related to gender, onset age, lifespan, neurodegeneration, and even the number of CAG trinucleotide repetition. For this, we use another technology that we developed, the BDASeq, that is an analytical technique developed by BioDecision to identify relevant clinical pathological features that be connected to our genetic data. Using this approach, as expected, and as well we know, one very important aspect of the disease is the number of CAG repeat length. Using this technology, we demonstrate that the longer the CAG repeat, the earlier the onset and that of age of our patients. And therefore, as expected, the less the onset, the less the death of age of our patients. This result demonstrates for us that the CAG is an important aspect of the disease. Based on this, for the first time, we identified target genes that are downregulated and target genes that are upregulated according to the CAG-related number of repetitions. This is very important because all patients with Huntington, according to your genetic exam or test have this information, the number of CAG repetition. So we can now understand what is the level of these specific biomarkers in these patients to infer, for example, prognosis, to infer the disease evolution in these patients and use in clinic, for example, as a potential biomarker. Using the BD Explore, we also noted that there is no gender difference from the onset age of the disease, as well very stated by the scientific literature along this data. However, we demonstrate that the higher the neurodegeneration, use one grade that is a scientific grade called von Settle grade. It's a scale that was used and very used in clinics to analyze this neurodegeneration, the level of neurodegeneration. And you observe that the higher this neurodegeneration process, the earlier the onset and age age, but larger the CAG repeat, demonstrating that the onset and that of age, as well the CAG, are important characteristics of our patients that be connected to our genetic data. Using this information, we identified for the first time novel brain degeneration related target genes. We identified genes that are downregulated and genes that are upregulated in the brain. And if you analyze the expression level of these biomarkers, we can infer how is the process of brain neurodegeneration. So we can use the same targets, for example, to monitoring the effect of one drug in one clinical trial, providing evidence if this drug have a potential to bring some benefits for our patients. Using the same approach, we also identified target genes that are closely related to onset age, identifying genes that once up or down regulated can be closely related to the onset age of the disease. It's a very important aspect of Huntington. We know that patients that develop the disease very early, and also we have patients that develop the disease with more than 6 years old. But today, we cannot understand why some patients develop the disease early or later. The only speculation and evidence that we have in literature for this reason is the number of CAG repeat lengths. However, this information, we don't have any biomarker related to this process. But here, we are demonstrating potential biomarkers that can be used to prove the efficacy of our drugs. But interestingly, we also identify genes that can be pharmacologically modified by several types of drugs, including drugs that are approved in the market for different diseases. And if you use these drugs, we can modify the expression of these genes specifically. And if you obtain this pharmacologically modification, we can increase the lifespan of our patients. So we can modify the natural history of the disease. That is what we are waiting for along all this time for all type of drugs in investigation. But unfortunately to date, there is no drug able to promote this effect. And there is no a consequence or of these drugs, but a limitation that the pharmaceutical industry have to develop new drugs. Here, we are providing these genes that can be used to develop new drugs. In this regard, we identify in these target genes that can be explored for the drug development or even for the drug reposition. This is very interesting. The potential drugs targets that we identify here in our study can be explored for pharmaceutical industry for two different things. The first one, to develop drugs, and second thing, to use drugs that are very common in the market and already approved by international regulatory agencies. And according to our study using Pharma intelligence, we demonstrate that it can be promote benefits for our patients. The main reason of this webinar is not indicate one specific drug. It's very important to remember for all of our patients that to improve the efficacy of these drugs that we are demonstrating in this graphic, we need to conduct clinical trials. We need to investigate with science the potential of this drug. But it's possible to infer that simple drugs, such for example, valproic acid, can modulate many genes, many pathways that are related to several steps of the disease and several aspects that lead to signs and symptoms of Huntington disorder. But all of this is not enough if you don't have one biomarker that can be measured in the blood. All these targets is very useful for us in terms of drug development because we are working with a brain disorder. But unfortunately, we cannot access these biomarkers easily accessing brain tissue samples. For this reason, it's very important to identify target genes to serve as biomarker that it can be easily measured using blood samples. For this reason, additionally to our study that investigate the brain, we also investigate blood samples of these patients. And we identified one potential biomarker. This is the first time in the literature that we are identifying one target gene or one biomarker with accuracy that it can be used to monitor the disease. To be considered a potential biomarker, it's important that this target gene, this biomarker, have been expressed in the blood, but also in the brain. It's important that this biomarker have been related to the signs and symptoms of the disease. And it's important to be deregulated in the brain and the blood. Interesting, note that this biomarker that we identified in the blood is among the 394 biomarkers or target genes that we identified as differentially expressed in the brain. We noted that this potential biomarker is downregulated in the brain of our patients, and this dysregulation or downregulation occur in a CAG repeat length dependent manner, making this a potential biomarker. But interestingly, this same biomarker that are closely related to oxidative stress that also relate to the disease and was responsible for neuroinflammation and was responsible for the neurodegeneration, it's increased in terms of expression levels in the blood of our patients. So here, we are providing a complete list of biomarkers and target genes that can serve for both drug design and even as a biomarker to monitor the benefits promoted for any type of investigational products in clinical trials. So, concluding, we can demonstrate that our technology is enough as very accurate to identify its potential drug targets. In terms of the targets identified in Huntington disease, our technique identified about 400 validated therapeutic and prognostic targets for Huntington disease, which serve as biomarker for other neurodegenerative disorders and aging-related process. Science, as we know, the Huntington disease naturally accelerates or exacerbates the aging process. So our results demonstrate and bring novel and useful therapeutic targets identified by our technique, bring hope for our patients. So from these 394 target genes, we have genes that are targets from brain generation that can be pharmacologically modified to increase the lifespan of our patients, that can be monitoring the blood of our patients to provide the efficacy of drugs in investigation, that can be related to onset age, and can be related to the CAG repeat length. All of these results have been noted in Brazil, Latin America, and world in several publications that are noted to demonstrate the potential of these results to bring hope for our patients. This is the main purpose of our study. I would like to emphasize that both our technology and the results using our technology and the results that were described in this presentation was awarded in the MENA Congress for Rare Disease. That is one of the most important congress dedicated to rare disease. To finish, I would like to thank my team, especially Dr. João Rafael Dias Pinto, who is with us in this presentation and was main responsible for the development of our algorithms and technology, and Benedito Falcinoni Neto, who is our statistician responsible for the development of our technology and validate all of our results. I would like to thank you, the organizations in Brazil and international organizations that support our study, such as International Huntington Association, Associação Brasil Huntington, and Federação Brasileira de Alzheimer, or Alzheimer Brazilian Federation. I would like to thank the support from the university that I am professor, Paulista School of Medicine from Federal University of São Paulo, FAPESP that is supporting us in financial terms, Google Cloud for startups, and Serve Information that is Google representing Latin America. I would also like to manifest my acknowledgement specifically for Dr. Luciana Munhoz, who is scientific coordinator in Brazil for groups of Huntington disease, and that have been discussing all this data with us since the beginning of the study. Professor Irina Kerkis from Butantan Institute, who is a leader of and scientist responsible for clinical trials for Huntington disease in Brazil, and with a long expertise in the disease with 3 decades of studies and investigational product development for Huntington. Dr. Joyce Macedo, neurologist and PI of clinical trials for Huntington in Brazil, and is co-author with us in our publication, first publication, and is our partner in this study, and Katia Favalli, that is coordinator of PESP and pharmaceutical that have been accompanying all this study since the beginning of this study. Thank you for this time. It will be a pleasure to answer any questions that you have and discuss all these results and details and complete access to the pharmacological and prognostic targets for AG. Please feel free to contact us by our email. Thank you so much.
Thank you, Professor Rodrigo. Let's start with the questions that we have here, and feel free for answering or asking for Professor João for answer as well. Well, but the first question we have here is from Croatia. Thanks for joining us, okay? How can this post-mortem data can help us with the treatment of a live patient?
Oh, perfect. Thank you for the question. It's very important to remember that we need to identify the target genes. When you are discussing about drug development, we need to understand how I can develop a drug if I don't know what is my target. So I need to access the main tissue or organ that are affected by the disease. As I previously informed, unfortunately, cannot access the brain samples using live individuals. For this reason, the only manner that we have to investigate these samples is collecting post-mortem samples. However, at the moment that we collect, this post-mortem sample have one procedure to be collected, and we analyze the quality of the sample to provide evidence that this sample has a quality enough to investigate. We are not talking about a dead tissue. We are discussing about one tissue that it collects fresh at only some hours after the patient death. And so we can investigate the same data that we can observe in a patient, in a live patient. For this reason, it's very important to access the samples to understand what are these genes or proteins that are modified according to disease, and with this, planning possible drug design.
Next question is from Guy Watson. Thanks for sending the question. How soon could it be possible to predict exactly one set for HD positive person?
All of these samples that was collected from individuals have a genetic test, sorry. So we have certainly that all of these patients are positive for the gene mutation. And we are investigating all of this information of these patients that are available in this SRI public data set. So we have certain about the mutation, we have the specific number of CAG repeat, and with this we can combine all this information to provide details about the relation of the target genes and the CAG repetition, for example, and the mutation level.
The next question is from Marcos Ferreira from the Brazilian Huntington Disease United Association. Which drugs you found to be used for GH treatment?
We have some potential drugs that are approved and can be repositioned from the Huntington treatment. Of course, it's very important to establish clinical trials to provide evidence of the benefits of all of these drugs. The main purpose of this study is identify the target genes. But also, we understand that pharmaceutical companies around the world have another database more sophisticated with molecules that are not publicly available for us, that have the potential to develop another and more specific drugs, including based on antibody treatments, biological drugs, and other therapeutics drugs. However, here we are evidence some potential drugs that can be repositioned for the treatment of the disease. One of the most interesting in our result is valproic acid. That is a drug with reduced cost for our patients. We know the effect of this drug a long time, is approved for the treatment of different types of psychiatric symptoms and diseases. Very well used in literature, used including for Huntington trials in the past. However, the investigation of these drugs was limited only in science and not in clinical trials. So we need to start urgently novel researches, novel clinical trials, and in this presentation, I suggest valproic acid. I think that is the main candidate for one potential drug development that can reduce the motor symptoms of the disease, but also can promote benefits for neuropsychiatric, cognitive symptoms of the disease. And in correct dose and posology maybe can lead to the postpone of the natural history of the disorder.
Okay, another question from Guy Watson. Is there value in studying brain fluid that occurs increased during symptoms to discover possible gene markers?
We can identify potential markers in each tissue that we think that is very useful for us. We can investigate here in this study using only brain samples and blood samples, but you can investigate in liquor, for example. However, in terms of the identification of these targets for us, it's very important to focus in the brain, because the brain provides us a list of potential drug targets, but focus in the blood because it's more easily to collect this sample tissue to analyze this biomarker, and with this to provide one evidence of drug investigations if these drugs have these potential benefits as we hope in our clinical trials.
Okay, another question from Croatia. How you think that can be used for a standard patient? What will be the outcome of this technology?
I think I cannot understand this question in terms of
maybe
what is a standard.
How can you use this technology, your technology that you developed, for the regular patient that has HD?
Oh, perfect. Sorry. It's very useful for all cases. I suggest that it's very important to refine all results all the time. We understand that human population has one heterogeneity that is very well known for us that are geneticists. So how many patients we can investigate? More specific are our targets. So what is very important? We need to perform an international study, for example, with live individuals, not with post-mortem samples, investigating the blood of these patients. We understand that the few number of samples collected and available in these public repositories are from blood. So if you can start one international study, we can use this technology not only to investigate our patients to discover novel potential therapeutic targets or even biomarkers, but we can use this information in clinic. We can, for example, use the genetic data for understand how the patient is to monitor our patient along the course of the disease. It's very practical for this. So the limits of our technology is not a limit to these post-mortem samples or even to identification of target genes for Huntington or for this study. We can apply this technology in other possibilities of studies.
Which some questions that I'm translating into English that comes from Brazil. What is the next step for your research?
Perfect. Thank you for this question. The main important aspect for the next step is to lead this information to the pharmaceutical companies and even to scientists around the world. I think that we have a very interesting scientific community in Huntington disorder. And it's very important to share all details, discuss with scientists, and refine all of these results. Here, we are providing a complete list for the first time of drug targets already validated, and also we are providing potential biomarkers that can be aggregated in clinical trials ongoing. So the main purpose of these results, and I think that next step, the most important next step, is to lead the results to pharmaceutical companies. We understand that as a scientist, as in universities or even in companies, as like our company, we are not a pharmaceutical company. We cannot develop one drug. We are providing information to pharmas, to big pharmas, to produce these drugs. This is our next step.
Another question that I'm translating right now. Your study could contribute to other rare diseases as well?
Yes. All of these results can be applied for all rare disease. When you're talking about rare disease, we have one limitation, the number of samples. We understand that Huntington disease, although it's a rare disease, we have an amount of patients in Brazil and several countries with the disease. However, we have some other diseases that are most rare than Huntington. And so it's very difficult to access these samples, to analyze and to collect these drug targets or biomarkers. The main potential of this technology and the combination of artificial intelligence and transcriptomic data analysis is that we can identify potential drug targets with reduced number of samples. We conduct here one analysis demonstrating that the same results, these results was not described in this presentation because it's not related to drug targets, but related to the symptoms and signs of the disease. We identified recently one important aspect of Huntington, for example, there are some deregulations that involve many other aspects of the Huntington disease that have been discussed along the 2 or 3 years. And interesting, we got the same result using only about 114 samples that another international study using 3,000 patients. This demonstrates for us that the main potential of this technique is that we can apply for rare disease using a reduced number of samples.
Another question. Why blood marker is so important and why it is difficult to find a blood marker?
Perfect. To find biomarker is one of the most important point of the science when you're talking about drug design or even when we're talking about clinical trials. From these 256 clinical trials ongoing or concluded that we have in clinicaltrial.gov, most of them only use one criteria to prove the potential benefits of these drugs, using a clinical scale for Huntington disease. However, this scale, although it's very important in a clinic for physicians, it's a very subjective scale because it's very possible to be manipulated. Even when you're talking about ethics in science and clinical trial, we hope that this type of questions is not evolved. We understand that the subjective of this scale could lead to misinterpretation. Moreover, if you analyze the clinical trials ongoing, you can observe that our pharmaceutical companies analyzing, for example, the motor score, other, the cognition score, other, investigating the chorea score. But what is the main aspect that we need to analyze? For this reason, it's very important to have one biomarker. What is a biomarker? It's a protein, it's a molecule that we can measure using one test. So there is no subjective in this type of analysis. We can collect one blood sample, we can measure the level of this protein, and you can correlate the level of this expression with the effect of our drug. For example, we are talking one simple question to answer this question. How you can prove that drugs for cholesterol treatment, for example, function? It's very simple. We administrate the drug for our patients, and we can measure the level of cholesterol in our patients. If our drug have one benefit, it's expected the level of cholesterol will reduce along the time. However, I can use a subject purpose for this. Imagine that I can prove for you that my drug for cholesterol is reducing because the patient is more happy. For example, I cannot use this information. Happen, not are direct correlate to cholesterol. Is analogy for the same case. Is subjective characteristic that I am analyzing here. I need to understand the levels of cholesterol. But in terms of Huntington, what is this level? What is protein that I need to correlate disease to prove the efficacy of my drug? There is no specific biomarker. Here today, we are using different types of approaches. For example, the levels of neurofilament protein that provide us with information about the neurodegeneration. However, the levels of these proteins are different if you compare, for example, in plasma, in blood, in saliva, or if you analyze in liquor, for example. But we don't have one specific biomarker for Huntington. Here, we demonstrate one potential biomarker. The main purpose of this biomarker is not complicate the clinical trials ongoing, but by contrast, it's offer for this company the possibility to prove the efficacy of their drugs. We have that many parts of the clinical trials for Huntington have many difficulties to approve their therapies because for regulatory agencies such as FDA in USA, in Brazil, Anvisa, or even EMA in Europe, don't like these subjective characteristics, such, for example, the scale for Huntington. These agents prefer one biomarker, one evidence that can be tested using biochemical assays. The protein that we identify in blood and brain of our patients that serve as a potential target can be measured using this type of techniques and can be used to demonstrate if our products are or not having these potential benefits.
Okay, thank you, Professor Rodrigo, for the webinar. I'd like to remember you all again that we have in 15 minutes another webinar for professionals, researchers, and pharmaceutical industry, which has more technical data. And the recording of this webinar will be available at the YouTube channel of Brazilian Huntington Association. Thank you all. See you later. And thanks for everyone that joined us today and sent some interesting questions. Thank you.
Thank you.